Hello, my name is Krista Mann, and today we're going to be talking about the interactions between Salmonella enterica and the epithelial cells of the intestinal wall. First, let's talk about Salmonella itself, specifically Salmonella enterica. Salmonella enterica is the leading foodborne pathogen in the United States and is responsible for typhoid fever and gastroenteritis, or the stomach flu. This bacteria is found in water, vegetation, soil, and the intestines, but Salmonella outbreaks in humans are mostly associated with contaminated eggs and poultry, and is mainly spread by improper hand washing and food handling. We're going to be talking about the interactions between Salmonella enterica and the intestinal epithelial cells, but before we get into that, let's take a look at the cell that we have in front of us. So we could see that this bacteria is 2 to 5 microns long and 0.5 to 1.5 microns wide. This is a prokaryotic bacterial cell, so we're going to have certain features like the nucleoid containing the DNA. You're going to have little ribosomes. You can see here that are 70S in size. And then, of course, you have your choice of gram-positive or gram-negative cell walls. And the Salmonella enterica has a gram-negative cell wall, so this is why this is pink, because in the gram stain, this bacteria shows out as pink because it is gram-negative. So you can see the different layers here. You have the plasma membrane, peptidoglycan, and the outer membrane. And also this cell does have a capsule. And then we see here that there are pili here. Like over here. And then you have the longer structures, which is the flagella, which helps the Salmonella enterica get to the epithelial cells, and we can get to that later. A characteristic about this cell that also helps it get to the intestinal cells is that it has acid tolerance genes, and this is essential because it has to get past the stomach, and the stomach is very acidic. Another thing to know is that this cell is facultatively anaerobic, which means that it can survive without oxygen present. So that makes it even more resilient. The last thing that we're going to note here is that this is a endotoxin producing bacteria. And I will explain more into detail why that is important once we get into the interaction between this cell and the intestinal epithelial cells. So here we got the intestinal epithelial cell. The top part is the intestinal lumen, and the bottom part is the lamina propria, the inside of the cell. And here we see that is the mucosa layer that I made orange, and we have the commensal bacteria, which I made green, which is just the normal bacteria, and this bacteria fights for nutrients and space against pathogenic bacteria like Salmonella that we see here. It is pink because of the gram-negative wall, and there is the flagella. And these flagella help it maneuver to that mucosa layer, well, through the mucosa layer to get to the epithelial cell wall. And the epithelium has many different cells fighting to maintain homeostasis and protect against pathogens like Salmonella. We see here that we have the M cells, Peyer's patches, the goblet cell, Panna cell, absorber cell, and the dendritic cells. So you see that the Peyer's patches are underneath the M cell, and beside is a dendritic cell because they all work together. And these cells work together to continuously sample the intestinal lumen to make sure they're are no pathogenic cells like Salmonella. So the Salmonella bacteria cell mainly targets the absorptive cells, M cells, Peyer's patches, or dendritic cells. And it does this by using the flagella to swim through the mucosa, attaching to the intestinal cell walls, using the pili that helps it kind of attach better. And once the salmonella interacts with the epithelial cells, it secretes toxins, which triggers the host cell, which results in the induction of membrane ruffles, which allows the salmonella cell to be engulfed by the host cell. And you can see that happening here. And that makes a salmonella containing vacuole where the salmonella cell can start to hyperreplicate, and that is how infection begins.